Lecture 6, the motor and somatosensory systems. But first, face area in IT. Degrees, no eyes, brush, no color. Fusiform face area, FFA, in inferior temporal lobe responds to images of faces in humans as well. Fusiform face areas, fMRI, when viewing images of faces. Special case of faces. Prosopagnosia. Prosopagnosia. Object agnosia, inability to recognize objects, includes visual agnosia, inability to recognize objects visually. Prosopagnosia, inability to recognize faces or see faces, face pareidolia. Oliver Sacks, best-selling author of Awakenings, The Man Who Mistook His Wife for a Hat, and other clinical tales. Prosopagnosia, face blindness, inability to identify or recognize faces, but no general deficit in object recognition, can occur as a result of bilateral temporal lobe damage. Fusiform face area far into the what pathway, but also congenital. Can recognize people based on other traits such as voice, gait, or other body features. Can often see emotions from facial expressions. Synesthesia, a condition in which stimulation of one sensory system results in unusual experiences in a second, unstimulated system. Grapheme color synesthesia is the most common. Numbers and letters have colors. Synesthesia is consistent and is now believed to be due to increased wiring between brain areas involved in processing different types of sensory stimuli. A test for synesthesia. Find all twos as soon as you can. Physical stimulus, what synesthete sees. What synesthete sees. Too many unpruned connections between V4 color and IT graphemes might explain color grapheme synesthesia. All the visual areas are interconnected in healthy brains. Graphemes, colors. Summary of visual pathways in brain: central sulcus, parietal cortex, MTV5, where, V1, V2, V3, V4, what, inferotemporal cortex, HCER, MV1, LGN, RGC. Lecture 6. Wilder Penfield, Canadian neurosurgeon, 1896 to 1976. 1940s, 50s, he stimulated the brain of awake patients to find source of epileptic seizures. Stimulating different parts of the brain elicited different responses, movements, sensations, memories, etc. Developed maps of motor and somatosensory cortices, homunculi. Somatotopy in motor and somatosensory cortices. motor cortex movement, electrical stimulation, somatosensory cortex, somatic sensation, swallowing, tongue, jaw, lips, face, eye, brow, neck, fingers, thumb, hand, wrist, elbow, arm, shoulder, trunk, hip, knee, ankle, toes, motor cortex, somatosensory cortex, tongue, jaw, teeth, gums, lips, face, nose, eye, thumb, fingers, hand, arm, shoulder, head, neck, trunk, hip, knee, leg, foot, toes, genitals. Somatotopic organization of M1 demonstrated with TMS. TMS equals transcranial magnetic stimulation used to activate neurons in the cerebral cortex. Magnetic field, TMS coil, electric current, skull. Somatosensory cortex, sense of touch, frontal lobe, central sulcus, post-central gyrus, somatosensory cortex areas 3, 1, and 2, parietal lobe, S1, S2, temporal lobe, peripheral reception, thalamus in coronal section, ventral posterior nucleus. S1 gets inputs from thalamus, represents info about touch, temperature, limb position, pain. S2 gets inputs from S1, higher order processing. 4 main types of somatosensory perception: fine touch and pressure, proprioception, perception of the location and movement of the body, nociception, perception of pain. Nociception, perception of pain. Temperature. The 4 types arise from distinct receptor cells in skin, muscles, tendons for mechanical force, heat, vibration, etc. Pain, touch, vibration, stretch. Somatosensory receptive fields, amplifier A, amplifier B, cortical cell responses, period of stimulation, neuron A, period of stimulation, neuron B, receptive field for cortical neuron A, receptive field for cortical neuron B. The receptive field of a somatosensory neuron is a region of the body surface where a stimulus will elicit action potentials in that neuron. Stimulus intensity encoded in frequency of action potentials. Longer or stronger stimuli release more neurotransmitter. Stimulus, transduction site, trigger zone, myelinated axon, cell body, axon terminal, amplitude, duration, membrane potential from -80 to 20, action potentials, threshold, moderate stimulus, longer and stronger stimulus, membrane potential, higher frequency of action potentials, more neurotransmitter released. Transduction stimulus, transduction site, trigger zone, myelinated axon, cell body, axon terminal. Measuring perception, 2-point threshold. 2-point threshold, millimeters. What is the minimum distance at which 2 stimuli, 2 simultaneous touches can be distinguished on your fingertip, on your forearm? Two-point discrimination test. Large receptive field, small receptive field. Compass with points separated by 20 millimeters. The receptive fields of 3 primary sensory neurons overlap to form one large secondary receptive field. 2-point discrimination test. Large receptive field, small receptive field. The receptive fields of 3 primary sensory neurons overlap to form one large secondary receptive field, skin surface. Compass with points separated by 20 millimeters. Primary sensory neurons, skin surface. When fewer neurons converge, secondary receptive fields are much smaller. Convergence of primary neurons allows simultaneous subthreshold. Convergence of primary neurons allows simultaneous subthreshold stimuli to sum at the secondary sensory neuron and initiate an action potential. Secondary sensory neurons. 2 stimuli that fall within the same secondary receptive field are perceived at a single point. 2 stimuli that fall within the same secondary receptive field are perceived as a single point because only one signal goes to the brain. Therefore, there is no 2-point discrimination. The 2 stimuli activate separate pathways to the brain. The 2 points are perceived as distinct stimuli, and hence there is 2-point discrimination. Similar to differential visual acuity of rods, high convergence versus cones, low convergence. Plasticity in the adult somatosensory cortex. A, 5 through 1. B, before differential stimulation. C, after differential stimulation. Digit 2 and 3 were stimulated with touch over and over. Phantom limbs. A phantom limb is the sensation that an amputated or missing limb, even an organ like the appendix, is still attached to the body and is moving with other body parts. Approximately 50 to 80% of individuals with an amputation experience phantom sensations in their amputated limb. Remapping of phantom limbs. Frontal lobe, temporal lobe, tongue, teeth, upper lip, elbow, arm, shoulder, leg, foot, toes, following arm amputation, somatosensory strip, somatosensory strip, following arm amputation, understanding phantom limb phenomena. One theory for phantom limb sensations is that the cortical rearrangements in S1 are somehow causing perceptions of the missing limb. Studies done by V.S. Ramachandran. Somatotopy in motor and somatosensory cortices. Motor cortex movement, electrical stimulation, somatosensory cortex, somatic sensation, swallowing, tongue, jaw, lips, face, eyebrow, neck, fingers, thumb, hand, wrist, elbow, arm, shoulder, trunk, hip, knee, ankle, toes, motor cortex, somatosensory cortex, tongue, jaw, teeth, gums, lips, face, nose, eye, thumb, fingers, hand, arm, shoulder, head, neck, trunk, hip, knee, leg, foot, toes, genitals. Does the map in the motor cortex represent muscles or movement? The motor cortex represents different types of organization, the part of the body that is to be moved, spatial location to which the movement is directed. Neurons in M1 also code for movement properties such as force, direction of movement, velocity of movement. Activity in M1 neuron starts before the movement, so they are involved in planning movement. Hierarchy of motor control systems. Initiation of movement, primary motor cortex, non-primary motor cortex, selection of movement, basal ganglia, thalamus, cerebellum, modulation, refinement of movement, brainstem, spinal cord, muscles of body, muscles of face, head, and neck. Deficits in motor system, damage to basal ganglia, like in Parkinson's disease, dyskinesia, tremor of the hands even at rest, persistent tremors, shuffling gait, unbalanced and in small steps, curved in a characteristic way, damage to cerebellum, ataxia, lack of coordination of voluntary movement. Deficits in motor system, damage to basal ganglia, like in Parkinson's disease, dyskinesia, damage to cerebellum, ataxia, lack of coordination of voluntary movement. Damage to basal ganglia, Parkinson's disease, is caused by progressive loss of dopaminergic Parkinson's disease is caused by progressive loss of dopaminergic. Parkinson's disease is caused by progressive loss of dopaminergic neurons, but damage in BG can also cause hyperkinetic disorders like Huntington's disease, excessive involuntary movement, opposite to dyskinesia. Huntington's disease is caused by a mutation in one gene, and it is characterized by chorea, involuntary and unpredictable body movements. Degeneration of a region within the basal ganglia causes more signaling to the motor cortex. There are no treatments available for Huntington's, but researchers are working on gene therapy and gene editing approaches.